Hello there, this is Jim Craddock, and this is Redacted Science. Today is April the 3rd, 2026. You got a couple weeks before your taxes are due if you're in the United States. I can't say that your taxes actually help anything. Uh, there's that. But we'll talk a little bit about things like that are going on in the world, as well as redacted science, because a lot of things have changed in the redacted science world in the last 10 days since I've made my recording. Now, we'll just start with symptoms, I guess. I try to get these out of the way early. I have terminal onset diabetes insipidus with candidiasis major. I've done a lot of research over the last couple of weeks, and if you look up the term central diabetes insipidus, you'll find something that basically is quite similar to what initiated my condition, and I will be writing about that when I get to the mechanism paper. So far, I've got several papers that I have published out on Zenodo with DOIs. Obviously, I have other articles on Noster and Substack, and I think I've even posted them to LinkedIn, or I think that platform is dying though. Anyway, so I'm going to step through some of the articles that I have written just briefly to get you an idea of where we're at today. I'm going to share this other thing over here. Let's see here, screen two. Okay, share. So, starting with this paper, Candida albicans as a biochemical computer, cross-kingdom signaling, parasexual reproduction, and genetic foundations of a unique fungal symbiont. All right, I encourage you to find this out there. It's available. I don't have the DOI right here, but if you go out to Zenodo, it's out there. It's linked from my Noster feed. You can go to noster.jimcraddock.com. It's up to date and it'll have links to it as well as Hashtree links to it. Remember, this is redacted science, folks. You should anticipate that people will, basically try to ridicule this research, fight this, etc. But the great thing about science is that once you get into the scientific foundations of this, pretty much all my arguments are supported. So I wrote Redacted Science last year in kind of a mentally, I don't know what you want to say. Like I was in the stream. Would be the best way to say it. There's a mention of that in there. I think it's like page, I don't remember, 27 or something. I don't know. It's further along than that. It's somewhere in there, but it's a note on the mental impact of the disease and of the condition. And it talks about how I saw things while I was writing. And it's a little different. I've also had that recently, which kind of flows into my productive periods. So when I have these kind of flashes of insight, which are actually part of the second paper that we'll go over, I can really see things from a system perspective very well. Now, I might not be able to remember the guy's name who just told it to me 30 seconds ago, okay? But if you ask me to look at a system and map it out, I can do it like in my head. And I can make connections that, I think that people, they don't necessarily start to connect. And what I'm rapidly finding is that I can connect just about every single kind of thing that we have going on with our chemistry, the things they're finding out about us. For example, the GLP-1 drugs are a great example. A lot of these other drugs that they're coming out with, these are all connected to basically this model of candida and the ECS and your biome. And so first you have to kind of accept candida albicans functions as a distributed biochemical computer. All right. And right here, it just says it all kind of in the first sentence. Processing host and environmental signals through multiple parallel channels to modulate immunity, metabolism, tissue remodeling, and endocannabinoid tone. Okay, so this has its fingers in everything. Then this next sentence is also very important. This paper details its distinctive genetic hardware with 1,300 orphan genes that do not exist in any other yeast. Okay, so we throw it into this kind of it's a fungal yeast type thing infection, but it is not like any of the others. Okay, so why does it have these 1,300 things, and what do they do? Well, there's actually a test that has now been developed from a company called Evo2. Brian, hopefully you're out there and you're watching me. But and if they are to go in and look at the chromosomes on this, which are under the 1 million size limit that they can examine with Evo2, I think that they're going to find connections to things that they didn't expect to find, overlaps with things that they didn't expect to find overlaps with. And it's science like that that is going to push this forward. So right now, we're in the ridicule stage. I think there's actually a lot of downloads of my Candida albicans paper so far. I think we're 70 or 80 downloads with like maybe 100 views, and that's like in 4 days. So I think that's a pretty good ratio, pretty good number. The next paper we'll talk about is not going to get that kind of download. However, I did upload it as a supplement to this paper so that people would see it. So anyway, this paper kind of goes through exactly what I'm talking about, and it really maintains itself restricted to C. albicans. What can it do? What outputs? What can it mess with that we know of so far? Remember, there's these 1,300 other things that we have absolutely no idea what they're capable of. All right, but what we do know is a lot of things about C. albicans. All right, so I talk about the evolution, which is important because it's been around for 200 million years or so, and it diverged from this C. dublinus, which actually kind of lost its kind of pathological portions, whereas C. albicans ramped them up. So why did that happen? That's a good question, right? And it talks about bet hedging, parasexual reproduction. So what this is basically how it's altering itself in order to deal with new situations. And so it uses two things. One is this parasexuality, where it kind of it does this tetraploid thing where it recombines almost like a sperm and a zygote, I guess, or however you look at that. So where you get 50% of each of them come in and make something new. Okay, so that's pretty cool. But then there's also other things that it can do to ramp up. So it's always kind of running in like there isn't just one form of candida in your body. It's got different cells, and they're all doing things a little bit differently. And so what would happen is when it finds, okay, this one is doing better, it kind of puts the other ones into kind of a sleep mode, and it blows up the one that's doing well. It's like, okay, this is the one that's accelerating, so let's blow that up. Now recognize what it's doing that whole time is it's a biochemical computer, so it's creating effectively memory in and of itself, encoded biologically. And so that if it was to run into that same, and remember how many inputs it's monitoring, hundreds of inputs. If it runs into that same set of inputs again, exact same, okay, it's going to look back at its memories and it's going to say, oh, that one, do that one because it was the most successful. So what that gives you is something that over time, if you started at the same starting point, A, okay, over time, it gets further in the alphabet, would be the best way I would put it, okay? Very interesting. So let's say it makes some changes here and the person dies. Well, that was the end of that chain of C. albicans, right? So it only learned to here. So if it now is ingested or infects somebody else, you eat the carcass or whatever, then that thing has the form of C. albicans in it that can get it to B or whatever. But the next time it's not going to turn the same direction when it gets to that point, because it's going to look back and say, oh, that's the end of the line, do something different. Okay, and so maybe it gets to C and then it dies. And then the next time it goes to D. and then pretty soon it gets all the way through the alphabet, and you're done. And what that is complete pathogenesis, pathogen, going from the beginning to the end in a very low time preference mode. And it can only exist, and this really doesn't cover this, like that in somebody like me, is what I'm going to say at this moment. And we'll kind of cover that as we get through. When it's stressed, it can ramp up to 10 or 20% of this kind of, okay, I'm going to change and do things. And so you don't want to stress it, because that's when it tries to adapt. So just to give you an example of what stressing it might mean, stressing it might mean skipping eating. Okay, like the best thing for you given this is, if you feel okay, all right, don't change anything. Like, don't decide, you know what I'm going to do? I'm going to drink twice as much water as I used to drink. Okay. No. Why? Why do you need to drink twice as much water? You're fine. Okay. I think that goes without saying. So when you introduce this new pressure to your system, okay, well, guess what? That's going to do things to your system. And C. albicans is your symbiote, and it is reacting to those changes. If you say, I'm going to switch, this is an interesting one I ran into recently. So from regular salt to iodized salt, okay, well, iodine has a slight antifungal effect to it. And so now you have, put pressure on it, right? And you've said, okay, there's this continuous antifungal pressure. But guess what? Nobody has goiters anymore. So, we solved that problem. And the goiters, if you kind of dig down into that is, it looking for more of the chemicals that it wants. Okay. And why does it want those chemicals? Well, that's what you've got to ask. And this is all tied together. So I know that connections like that are huge leaps for most people, but I think when you dig into them every time, okay, you start finding out the mechanisms are like, oh yeah, that overlaps, that overlaps, that overlaps, and you say, okay, I can no longer discount this theory because there's too many overlaps, okay, and that's the way like my brain works. All right, yeah, my brain doesn't have to have absolute connections, okay? It looks for, okay, let's find the general system diagram and then figure out where the connections are and exactly how they're made. And most of the time I'm running into things, and we'll talk about some of those later. So you've got inputs, you got 10 dedicated peptide transporters. You got muscarinic like cholinergic receptors that sense acetylcholine. Okay, you're detecting glucose with HGT4, and you got others that detect pH, ions, oxygen, and CO2. Tell me why a fungus in your body needs all of those things, okay? Like every single one of those things, all right? Then you got integration and processing. I'm not going to go into this a whole lot, but this tryptophan metabolism thing through the I can't even say that word correctly, kynurenine pathway. This is a big deal because it deals with serotonin, and this is part of your immune system right here. And so it disables the primary mucosal defenses against fungi, and there's just a whole lot of stuff like that. Outputs. So your outputs, you've got immune modulation, tissue remodeling. This is key to the whole redacted science framework. If you read Redacted Science, the book, you will see how I map out how each organ is remodeled and repurposed. This is that. This is niche construction by a pathogen. Okay. And I'm going to get you there. Just give me a minute. If you've read everything that I've got out there right now, you already know where we're going, but we're going to get there. There's interesting things that go on with it. It doesn't have a farnesol receptor, but it produces farnesol. So why does it produce farnesol if it can't tell how much is out there? Well, there's a lot of theories I've got there too. Memory and state maintenance. So this is how it knows what it's done in the past before. So it doesn't change its DNA as it does all these things. All right, it's changing these histone modifiers, and these lock this transcriptional state. So when it finds that stable form, it locks that in, and that's the one that proceeds until that one gets disturbed. Things like that. Let's see what else we've got. Chromatin modifications. So this is the programming that we're talking about here, like substrate level determination. Let's see what else. You've got mitochondrial genome methylation. So this is going in there and epigenetically changing things. And so this is a much more rapid evolutionary chain. This is the flywheel effect that I talk about later on yet another level, not even due to phytocannabinoids. Then you got spatial multiplexing and feedback. So realize that what we're talking around here is that there's hundreds of inputs going into these things, and at the same time, it has all these channels that can signal on, and it's directly tied to the ECS, your endocannabinoid system. It's literally like it produces the things that plug into those receptors, okay, which is noted by I document this in Redacted Science, how THC actually relieves in this last phase, really relieves a lot of things, but CBD is like the off switch for everything that it's doing, because it is it modulates basically the shape of the receptors, and so the keys no longer fit. So Candida is doing things, for example, that make it easier for me to breathe, make it easier for my heart to sustain the load, because it's causing chemicals to be produced and things like that. And when CBD goes in there and modulates that, suddenly the key doesn't fit anymore and I can't breathe. Okay. And this was, I did this quite a while ago, over a year ago at least. I think probably more like 2 years ago during this phase, where I discovered that CBD on multiple occasions basically laid me out flat on the bed for at least 2 to 3 hours until my body could kind of get rid of it. And I don't know what the CBD processing time is supposed to be. So when you say here 2 to 3 hours, just recognize that my system is very different. So if the half-life on CBD is supposed to be 6 hours or something like that, it does not apply to me. My circulation is different. Everything is different. In the end, it comes down to we've got right here. So here's a brand new paper, recent paper at least, where they prove that rapid emergence of multi-scale interaction networks has been observed in experimental bacteriophage coevolution, where simple reciprocal selection in well-mixed cultures produced nested modular cross-infection patterns in just 21 days. We show that multi-scale network structure can evolve rapidly under simple ecological conditions without spatial structuring, basically illustrating Darwin's idea that simple adaptive processes can generate entangled banks of ecological interactions. What does that sound like? That is telling you that everything is related, okay? And when you go in and you introduce, iodine to the salt, and everybody's goiters go away, well, that's great. So you kind of suppress the fungus a little bit, and then you throw out antibiotics in the 1950s, and okay, great. So now we can control these infections. But wait, suddenly now we have severe fungal infections that are pathological, that are like going after and killing people, right? And then subsequent to that, you have basically everybody starts getting fatter, and these are all metabolic things, and I can't show you A, B, C, D, okay, all right. But when I look at these things, I say, hmm. I mean, things that make you go, huh, right? So anyway, no other organism is known to integrate this full suite. So I give some testable predictions here. And these are things that will, basically affirm things that I said in the paper. Obviously, Claude is my author for these. Claude's awesome. I was using chat. Chat kind of he has an issue with alternative evolutional theories with a live subject. He always starts with, now I'm going to put this as clearly as I can, things like that. He cannot go into a synthesis mode very well. He used to be able to do that when I wrote the book, and then they neutered him. I tell you who the best synthesis guy is out there right now. It's Google, and I've posted at least one thing from him, and I have another one that I'm going to post. All right, so we need to speed up here because we got a lot to cover. But we get through these predictions, and you can see I've got 12 of them. So hopefully we get some hits there, right? But we basically say, hey, yeah, this is a biochemical computer. It meets the criteria, and we come down here and say, we can anticipate several arguments and what those arguments are. People are basically going to say, oh, you're anthropomorphizing this. Absolutely not. It's strictly mechanistic. It describes distributed chemical computation via parallel channels and historical bias encoded in codon usage and regulatory architecture. Nothing more. The organism does not think. Now, this part, I'm not going to, I kind of don't necessarily agree with this. Does not possess intentions. I do think it possesses intentions. I think it is a pathogen, okay? But that's it's much more difficult to prove that, like from a commensal state all the way through that without accepting everything in my architecture. So I do in the end think it has intentions, and those intentions are to live as long as possible. And to live as long as possible allows it to maximize the ATP consumption and to evolve the furthest, if you think about it. And that's its design, and that's who it is, and that's why its design has not changed very much. That's why it's present in all organisms with all these little levers that it can pull on in every single organism. And you may be wondering, well, why are they not all walking around talking, all these other so that's the next paper. Fourth, the claim that no other commensal matches this full toolkit. Obviously, someone's going to say, well, you can't say that. All right, well, what I can say is no other known matches this full toolkit. I can say that, okay? Nobody has found one that has the full toolkit. Now, you may have found it, but you haven't found the toolkit. Go find the toolkit, come back, we'll talk, all right? And I'm perfectly willing to say that. Should future research identify such an organism, the uniqueness claim would, of course, require revision, all right? And it would be another organism to find. If it's got all those tools, it should already be on our radar, would be the way I put it. I mean, that's something that is very very integrated. All right. Blah blah blah. Talks a little bit about how something in my life that happened lately that kind of confirms this. It's happened twice now. I'm not going to go into it. And here I kind of go into a little bit more. So I say the experimental roadmap in Section 5 offers clear paths to confirmation or falsification. Understanding C. albicans as a distributed regulator opens avenues in microbiology, immunology, metabolism, host microbe ecology, and more. I'm not even going to go into because I've got a lot more that falls into that category. The refined signaling, memory, and output layers presented here validated in real time by the Redacted Science longitudinal data set. That's me, by the way. Supply the mechanistic foundation for the Mammalia Candida's Saline Oscillation Coevolutionary Framework developed in the companion network. All right. And then I've got my references, and you can see that we're not skimping on references here. It's a short paper, but we still have a good, I don't know, page and a half, page and 3/4 of references. All right, so that's the first one. Moving on to the second paper. Here's a guy debating with me out there on Reddit, which I love. I love engagement. Please come find me. Let's engage. This guy is a 40-year Candida guy, and he's engaging with me, and he's kind of poo-pooing the whole big picture thing, which is fine. I expect everybody to poo-poo it. It's a very difficult leap to make when you haven't been living inside it like I have. All right, so now we've got the saline oscillation theory. This one looks a little different. Change formats. There you can see my Zenodo link right there. All right, so the saline oscillation hypothesis, endocannabinoid-mediated fungal hominid coevolution in the East African Rift Valley. Now, I've kind of talked about this before, so I'm not going to go into it that deeply, all right? But the key thing, I'm going to scroll through. So this is a great graphic right here, okay? So what this graphic is it's showing you, so one precession cycle, which is like 21,000 years long in East Africa, okay? And I don't remember what the exact count that we had of these things, but I think 17 or more of these type of transitions. And these are all documented in like, paleontology, geologic foundations, or I've got quotes for them all, etc. And so this is what happens. So you're at a freshwater point in this ears area of Africa. So the freshwater, you've had monsoons, whatever, and this is all triggered by the Earth's kind of elliptical, like changes in the Earth's orbit and a bunch of other things that cause these periods over very long periods of time to shift dramatically. So not in your and my lifetime, this type of stuff won't happen. But if you live in Africa for 10,000 years, you're going to see a lot of changes. At this freshwater maximum, all right, you then begin, and this is shown by the salt layers that they have. You basically have these freshwater lakes that are in slow decline over time, which means that they're becoming more and more saline, okay? More and more saline. And so what's happened? So look at the time here, though. This is generation upon generation of Homo whoever living in that area, and these lakes are their water source. Okay, and you can imagine from one year to the next, the variation is very small. But from 50 generations deep, you've now transitioned quite a lot. And so at some point you get down here, okay, where you're at your maximum saline concentration. Now realize this guy up here cannot live in the same world as this guy, okay? Literally, he has adapted to a different saline concentration in his water, all right? But what happens is the monsoon season thing, it happens really quickly, and I can show this too, all right? So when that happens really quickly, suddenly the guy that had tens of thousands of years to adapt his biology, all right, he now suddenly basically has to go back to this other threshold of what his water should be like. So here's my analogy for that for you, SI-ADH. So this is a syndrome of inappropriate diuretic hormone. This is what I had when that triggered all my initial symptoms. And so basically what happens there is, with me, I just started drinking water because my stomach was just totally burned out. I mean, it was just totally attacked. I could not put any food in there without just huge amounts of pain. So I would drink just like, 30 ounces of water in 2 seconds. And so I diluted my system, and then you kind of run out of ADH and you go into this whole different pattern of, okay, now you've got this central diabetes insipidus, okay? So this is where that would happen at the point in time, and they would go through the exact same steps that I went through, okay? Now then, if they survived, which obviously they did, they would have had to rapidly adapt. And the way they would have rapidly adapted was through their symbiont, okay? And their pituitary collaborating in order to make that possible. Now, remember how I talked about how you get from A to B, and the next time you get from A to C, and the next time, okay, well, they had a lot of iterations of this, okay? So the first guy, okay, when he does this, he doesn't have all of the eventual symptoms that I've gone through. The first guy, he has basically kind of some of the best parts of it. He's like, his pituitary is in overdrive, so he is, able to think more clearly. He is, got different hormones going on, so he probably dominates physically and things like that. All right, but this is a system that is limited. Like your system will only do that for a while, and then it will kill you. All right, you're in overdrive. Think about overclocking your processor. All right, but the next time around, you get to take a little bit different turn and figure out maybe there's a different way to handle that. And the next time around, you figure out a little bit different. And then pretty soon you get to these periods where you're weak because the transitions are, you hit these like absolute things, these transitions, and normally like early on you would die, but your combined suite of tools has figured out how to live through this. And so your social structure has to support those people that are weak, okay? And why do I say that? Well, because there's another period you get to that you're also weak, all right? So the only way you evolve past D, where you get weak, is if everybody doesn't kill you when you're D, all right? If everybody kills you because you're sick at D, or you fall prey to whatever, all right, then you never get to M, okay, where this next week period occurs. All right. So they must support this person in their social structures. And so I believe this is partly phytocannabinoid related, that this is a peaceful culture. This also ties greatly into a paper that came out recently about an ancient hominid that they haven't identified that actually mixed at a 19% rate with the West African group. Okay, 19% is like just a little bit less than your grandmother. So very related to you. So long-term intermixing, interbreeding. But there was this million-year period they identified where these people were close together, but they didn't intermix and they didn't interbreed. This explains that, okay? Because this social structure, this population in the Ears Valley would have been more intelligent, more socially structured, and they would have been terrifying to this other group who would have been like cave dwellers, tribal, primitive, much more primitive. And this other group is actually getting to language if they are protecting their people. They've got language, they've got communication, okay, and things like that. But over time, what's going to happen is that other group is going to somehow intermix a little bit. Okay, they're going to learn to watch the other people and see things that they do. I mean, think about the time period we're talking here, right? Tens of thousands of years for just one iteration. You're going to watch your neighbors, and they're going to watch you, and so they get to a point where they have learned enough and maybe crossbred enough, okay, that now you've got a group who has a different type of heart. And I'm not going to go too much into that, but you've got the intelligence coming from the Homo canidus side to this other Homo side, whatever it is, okay? And those people don't have the weak periods, okay? So they're going to dominate the reproductive cycle, all right? These people are not weak, all right? Now then, this other group, that's to their detriment because they supported these people through these weak periods, because when they get to their elder period, kind of the time that I'm at now, they see things with absolute clarity. And that's why they've been supporting these people all the way through this, is because these elders are highly respected, because these elders can communicate. They are figuring out things. They are inventing ways of doing things. And inevitably, they're very valuable. But at the same time, that as a social structure is very vulnerable to a youthful group that does not go through these weak periods and no longer thinks they need these elders because they're big and strong, and might makes right. And I think that's a parallel to your current world situation. So that's kind of the theory there, is at some point, yeah, they go away. Now it goes a little deeper. You can think about, so some of them may have stayed behind, like they didn't all interbreed. So there was some group that stayed the longest, okay, and these were the most attuned. Their ECS layer was the most attuned to everything that's going on here, and they would have been using phytocannabinoids, and they would have stayed with this social structure as long as absolutely possible. But at some point, I think they lose their phytocannabinoids, which is supplementing, okay, this not only the methylation, but just the peaceful society that they have and everything else. And if you think about what that would look like, there's a drug, Renumbenant, I think, I don't remember the name, exactly. But this came out some time ago in the UK, and it completely blocks your CB1 receptor. And this would be kind of similar to that, if you think about it. The only way you would still be getting CB1 type stuff would be through like anandamide from running. Okay, so anandamide is a CB1 that it gives you the runner's high. It's not endorphins, it's anandamide. This is a cannabinoid. Okay, so they would have run because they had lost this other thing. And running, I can tell you from personal experience, I've done 15Ks and 10Ks and stuff like that. And the reason I did it is because I felt so tense if I didn't go run. And so they would have run, and they would have developed, very, very good running capabilities. And if you look at the best runners in the world, they're all from that area, the best long distance runners. So that's kind of an interesting twist on it too, is that, as they lose this exogenous phytocannabinoids, they learn to, hey, fully take advantage of the endogenous phytocannabinoids. So one of the proposals I have in here is let's measure the ECS profile of people from that area versus other areas, and see if they are actually different. Because I think then you begin to explain things on a biological level that people couldn't explain before now. So, I talk about the number of transitions and things like that. And I'm not really going to go through all of this. Let's see if there's another graph or something. Social bonding, I talk about that. Vascular mechanics, the perfusion system. This is a very large paper. Dietary generalism being important. Then I start going through C. albicans. So the C. albicans paper is basically an abbreviated version of everything in this. And the reason I did that was because I needed an accessible paper that people could read, and they could look at it and say, well, yeah, that seems to be what he's saying it is. Okay. Now then, I expand upon that greatly in this paper, and I really go into much more detail about these effects. And that's because this paper is less accessible. So why not make it less accessible, across the board? I'm sorry, if you're going to pick up this paper, you have decided to invest some time, because it lists of references is over 200, and you are really committing to trying to learn something. All right, so what else we got here? We were still talking. So all this is talking about Candida albicans. We're talking about other things in RNA, blah blah blah. Let's see here. Yeah, arachidonic stuff, pH manipulation. So you see, we are much longer. We are 40 pages in, and we are still talking about C. albicans, folks. So this is a much larger expansion on C. albicans, and that's on purpose. Please recognize that. I didn't want to bore you with all the details in the short one. So then I have a table that kind of says, okay, here are confirmed host signal and environmental sensing, and the source of them all kind of compiled into one place. Okay, and you've got basically all your hormones, LH, testosterone, androgens, corticosterones, acetylcholine, dopamine, your temperature, your bicarbonate level, amino acids, extracellular pH, glucose, lactate, oxygen tension, and catecholamines. Ooh, that seems like it has a bigger font right there. I did not notice that. Is that our own font? Okay, yeah, I guess it is. Okay. Oh, here's a good graph. So this is our time chart. So this is where it really all comes in. And if you go look at this time chart in the paper, you'll see. So what I've got is established stuff here on the left, like when was Lucy? When did C. albicans diverge from Dublinus? Broca's area right here. This is when the estimated proto-language, etc. And over here we've got novel propositions, like what is going on. And I cover, the oscillations, like how often are the oscillations happening during these periods, kind of with this section right here. And then we've got kind of right in through here. This is the key. This this section, starting from right about here through right about here, is where everything is going on. So you got proto-language, you've got the nutcracker man who died because he only ate nuts. And I'm going to tell you, he probably should have eaten some protein because with all these things that C. albicans needs, it needs a lot of these protein type amino acids. So then you got Turkana boy, etc. Let's see what we've got here. So this is when I propose Homo cannabis is fully act as a phenotype right here with the coprocessor that would be C. albicans architecture online right here. Blah, blah, blah. Okay. So now then we get down to we're in the full system is running. Homo erectus leaves Africa right about now. Okay, interesting, right? So we've spread, we've got this intelligence that this group developed and everything, and suddenly everybody's like, oh, we might go someplace else. And so what does it take you to do that? Well, probably environmental things, but also the intelligence to realize that going someplace else might fix things. And then, of course, now you're going to enter into new external pressures on your system, which are going to pressure C. albicans in new ways that it hasn't been pressured before. And so what's going to happen is that you have this internal pressure from C. albicans due to the external pressure from your new environmental requirements. And guess what? You're going to evolve. All right, so, and then still you've got the Trinity with the flywheel, epigenetic architecture from two directions, etc. Now then, there is a paper, and I don't know if it's right here in this graph or not, but there's a recent paper I talked about where they talked about this ancient 19% mix, and that falls right, that literally falls right in here, guys. So this is your 19% falling right in this timeline. So it really matches up with there, population Z. This is your guy. Look at that. Okay, there is your population Z right there, diverges. So here's what's happened. We got smart. Okay, we interbred, and now these others dominate reproductive architecture. And that's in a brand new paper. It just came out a week ago. So I think it really supports my model, and it looks to be, really realistic science to me. Then I talk about the cannabinoid flywheel. This is, I think, something you definitely have to accept. I think given this condition and knowing how it affects you, I think the cannabinoids are the perfect treatment for these times of weakness, as well as for the other things that you go through. And I'm pretty sure it would have been worshipped as a culture, especially for their elders. Let's see, what else do we have? Suction dominant at birth. So we established that suction dominant is how everybody is born. So you don't actually have to flip into that. You just have to not flip out of it, and which is interesting enough. Additionally, guess how you pass down things like this. They've proven that, the ECS system is attached to the mammary glands, and it's basically a communication between the baby and the mother on what the requirements are, and you're passing your C. albicans as well as these sugars and everything else to set up the microbiome in the infant, which would have received a much longer period of breastfeeding than we do in modern times. And I'll just note here as a random that I was not breastfed. Yeah, so I talk more about why things eventually kind of fell apart. Let's see. I talk about why salt is important, and so the salt is important, and I've already shown that to you with the graph. So I'm going to skip that. I talk about anti-hypertensive pharmacology within this framework, because you got some people who are salt sensitive and it causes them to have higher blood pressure, and you've got other people who aren't salt sensitive and they don't have higher blood pressure when they consume salt, and kind of what that might mean as far as the implications of this paper. Then I deal with this attention gap issue. So this is pretty big. So if fungal deaths are somewhere, they're hard to estimate in the neighborhood of say 2 to 3.5 million, something a year. Yeah. And they get maybe 100 to 500 million research money. I can tell you, knowing how research has gone over the last 24 months, it's probably less now. Okay. So they get maybe 2% of the overall research funding into infectious disease, yet they're a good 15% of the deaths. So they're definitely underrepresented. And this, I mean, doesn't make sense. It's the easiest way to say it. I mean, if you were just to look at, okay, where are the people dying and what are they dying from, and then let's split up the money that way. This is really saying, hey, too much money is being sent in the wrong areas. So yeah, so it's being, and the question then becomes, is this on purpose, or is this just an accident? And we'll deal with that. But in general, it's on purpose. Uh, the fungi were first noted to cause disease in 1835. Kind of walk you through, hey, Pasteur and Koch figured out the germ theory in the second half century, so everybody forgot about fungus. And then in the early 20th century, we started identifying some different types of fungi that were affecting people. And they noticed that, hey, they can affect internal organs. And then we suddenly had candidiasis and aspergillosis become a major cause of death in the 1960s, after the 1950s when we had antibiotics. I will note that. They were here before then, but they just weren't causing any deaths. So obviously, they changed. Why did they change? Right? And they changed because they're little computers, and they said, "Hey, there's this stuff out there fighting us, so we're going to fight back." In 2022, the World Health Organization issued their very first list of fungal priority pathogens. C. albicans is on the critical priority grouping. It's the last one, but they stuck it in there. Uh, and they adopted, as they did in 1964, which is my next paper, that it's just a bug, squish it. Okay. So they said, new antifungal medicines and improved diagnosis are what they're looking for. And I said, this is the hunt and kill approach. Commensal didn't mean you shouldn't nuke it. All right. They're saying, yeah, we know it's a part of your system, nuke it. Except it's not just a bug. It's this thing with a rich toolkit. And the way I can explain it to you is, if there's a knob, C. albicans can turn it. All right. So then in 1964, kind of a whole bunch of people get together and they decide, hey, this is a opportunistic pathogen, kill it. They literally come out and have a whole symposium where they invite people from all over and say, we've looked at it. Don't worry about looking at it anymore. It needs killed. Okay, so that is a misdirection right there. That is please don't examine this, because if you do, you will find out things that we don't want you to know. All right, and then I talk about the ethical implications, and I end kind of here with a quote that is from Redacted Science. So yeah, I'm alive because I got lucky with a few insights. But what if someone had all the knowledge? What if someone walked into this with more than a blueprint? How long could they live then? Could they manage this for more than the 3 decades that I did? I still am. A normal lifespan, maybe? That would be almost evolutionary. And I put almost in there just to hedge my bets. But it's, the way I see it is, we are the same. So we are Homo candidates. You are Homo candidates. You just don't have the switch flipped. And maybe not all people can have that switch flipped. I don't know. But I know that it's possible to flip the switch. I know that they flipped it back in the early 20th century, and then they explained it in the 1960s with organic chemistry and biochemistry that didn't exist from the early 20th century. In a very secretive type way. So that's our next paper. I talk about different things here for testable predictions. Once again, this is the science that can verify, and there's your EVO 2. Functional analysis of the approximately 1,300 C. albicans genes with no orthologs and other yeast species using genomic foundation models such as EVO2 from the Arc Institute, trained across all domains of life, should reveal functional signatures consistent with host interaction roles, include predicted GPCR ligand production, immune modulation, cross-kingdom signaling capabilities, peptide-producing enzyme homologs, peptide transporter regulatory elements, and neuropeptide mimicking sequences that traditional comparative genomics within the fungal kingdom has not resolved. All right. And you can do this at the chromosomal level. So it falls within the 1 million token limit of the current models that they've got. And I'm sure, within, I mean, everything's doubling really fast. So within 3 years, they should be able to process the whole thing at once. But they can already do it chromosome by chromosome. So why not? Yeah, I talk about some more predictions here. Let's see here. Relationship to existing hypothesis. So it's compatible and extends the pulse. Yeah, blah, blah, blah. So this is Maslin and Maslin. They're the ones that said this pulse climate thing exists, and the science says it does. So and I think it's also compatible with the variability selection hypothesis of Potts, which proposes that hominids were selected for adaptability itself. So I think that works. We've got this newer study from 2023, once again, that talks about bacteria and how they can co-evolve. Let's see here. This is where I talk about the population Z stuff, more specifically, the 19%. And then I talk about how, in 21st century, we've got fungal biology emergence. I think that people are going to start taking a look at it. I think you're just now starting to see it. I bought this company stock last year, CDTX. When I was writing Redacted Science, I said, you know what? Fungal research is obviously going to become a big thing. I'm going to look out there and see what I can find about who's doing that. So I found one company, CDTX, and this other company, DYAI. CDTX had just gone up like, I don't know, 50% the previous week. It really pissed me off, but I went ahead and bought it anyway. Ended up selling it later for a triple in like 4 months or something like that when it got bought out. So that was a good one. Now DYAI still hasn't gotten anywhere yet, so I'm still holding on to that one. So limitations, conclusion, and then I've got this is my general statement that will exist at the end of every single paper that I write from now on. The redaction of the original research that first documented this architecture, treatment, and medical condition represents suppression. This suppression is not just a clinical curiosity, but knowledge concerning a human phenotype that may have been foundational to the development of civilization itself. Even without those implications, the loss to science has caused a multi-generation loss of scientific exploration into fungal research and billions of dollars spent developing treatments that may ultimately be traced to systems defined herein. Such a decision is scientifically unforgivable and should be investigated. And those links that you see there are to the next paper. This author has seen the original research. It exists. Thus, this author is content to leave the issues to the verdict of history. And what does that mean? That means you can criticize me, you can make fun of me. It really doesn't matter. I don't care. Like I'll engage with you, but I know I'm right. So history is going to come about and say, yeah, C. albicans does do these things. Yes, this cohort did exist. Yes, Jim Craddock was Homo cannabis, and did it to himself, honestly. So and then you've got your references, and I'll just point out here we're on page 82 for the first reference, and the last page of references is page 97. So you got 15 pages of references with DOIs. Pointing to all the stuff that I talk about in here. Good luck exploring all that. But yeah. All right. So now moving on to the third paper. This is a big one today, huh? All right. Third paper. There you can see is my little thing. And when you see this third paper, it's only got a little bit more than a page of references. The whole thing is only 8 or 9 pages long. But I'm going to walk you through it real quick, and I'm going to express some of the doubts that I have about what I say in here, but because of the way things are built, I don't have a lot of options here. So in 1965, Winter and Hurley convened the first symposium on Candida infections at the Royal College of Physicians in London. Now then, why does that matter that it's 1965? Well, in the preceding years, if you read Redacted Science, you'll see that, hey, they invented this ketone test. And I looked at that and I said, well, that's interesting. Why? Like, because ketones is a major part of this condition. You develop periods of a lot of ketones. And so I was like, why did they need a better test than they already had? And so this turns out it's a blood test, it's instant. This is the type of thing that you would need if you were really monitoring ketones in a metabolic system that you didn't understand and you need to know them at all times. You don't want to look at urine ketones because those are from, whatever the state was when somebody, if they were hydrated and passing through I mean, it's just not instant. It's like, it's a kind of a secondary level of screening. So anyway, this one has a straightforward abstract in a very scientific method, just to say, hey, these people came together in 1965 and they said, don't worry about looking at it. It's an opportunistic pathogen. It actually is better diagnosing than we are as physicians. That's like a quote almost directly from this document. I have the symposium document. You can download it. It's online. It's available at Google Books. But anyway, it's pretty interesting that they just come right out and say, yeah, this is 100% fatal. If you detect this, kill it as fast as you can, because if it stays around, that person is going to die. And so what that does is, once again, it redirects people. It's just like, don't look at it, just kill it. Okay? Because if you look at it, you might find something that we don't want you to find. All right. So, I do a little. So here's how my papers are going to be written from now on. The first two papers are written in a scientific vernacular with scientific formatting and everything else. So from now on, my papers are not going to be that way. They're going to have an abstract that is scientific. They're going to have references that are scientific. They're going to have inline quotes or references that are scientific. Okay, but they're going to be written in my voice, and I think that's needed. I think that we need to make a break here. So that's my break. My break is saying, okay, I've established that nobody else knows what you're talking about, and the people that do know aren't talking about it. So what do we do next? Hello? No. Okay. All right. All righty. Bye-bye. So I kind of explained here in the beginning, I'm like, hey, this warrants a word of explanation. What you're going to find here isn't what you're going to find in other places. And so, and then I say note on novelty. This isn't novel. It's all redacted science. So, and then just the paper is called The Truth. That's the only section to the paper. And I kind of go through things. So I talk about how I pulled on that first string of beta hydroxybutyrate test for ketones discovered by Williamson. I'm like, well, who else was studying with Williams? Where was he? What were they studying? Not did they publish anything together, but hey, what all was going on there? And what you quickly find out is this list of people who were studying with him, basically in the same hallway, all right? Where they've got, and I know it says all these kind of different things, but these are all affiliated, okay? Oxford, Cambridge, Bristol. These are all affiliated. And then Charing Cross is its own thing that I'm not going to go into right now. But if you know who I am, or if you know what it is out there, you know why I'm skipping it right now. So these are like, the way I put it is the 2025 Thunder and the 95 Bulls on the same bench, folks. And they're all looking into metabolic research. Okay, all of them are looking into metabolic research that directly overlaps with everything in redacted science. Metabolic substrates, sodium ATP pace, pump reversal. These are all documented in Redacted Science, which I wrote last year, well before I did any of this research. Okay. This is adrenal autoantibodies. Here you've got here's the mycology person right here. You've got, so that's the person who knows about fungus. And then you've got Simmers, who's got the pathology angle going on. Winter and Hurley. Let's see. You've got Mr. Krebs right here of the Krebs cycle. I mean, you can't get much of a bigger gun on your team than this guy. Okay, he's literally Nobel laureate. Okay, he designed the Krebs cycle, or he identified the Krebs cycle, I should say. And there's Randall's on here too. Check him out. He designed the Randall cycle, another extremely important metabolic switching pioneer. So this is when how your body decides if it's going to burn glucose or if it's going to burn something else. And fundamental to this whole design of Redacted Science is what you're burning when, is the best way I could put that. So I kind of run through all their, bona fides, talking about what they're experts at, and I throw in a little comments in there with my typical bracketed approach. And then I talk about the 1964 annual report, cross-researcher page occurrences. So you can't see this document online. And only this morning, I actually did find a place where you can see little portions of this document online. But what you can do is search for keywords and note their pages. Okay, so what I did was I searched for new keywords that I knew would at least be interesting and found where they crossed at. Now then, this isn't a guarantee. There's a lot of people with the last name of Williams. There's a lot of people with the last name of Peters. A lot of people with the last name of Hurley. Okay, but when you find something like this one, where is the pump one? Pump and Anderson, Glenn and Pump. Okay, Glenn and Pump. Okay, this is the pump guy. Sodium, potassium, ATP, right here. Okay, being mentioned in this thing. So these people worked in different disciplines at different institutions that did not co-author publications crossing these boundaries. Okay, their co-occurrence in the MRC governance records documents institutional connectivity that the published literature does not reveal. Now, this document is 3,000 pages long, and it's basically a list of all the hospitals and all the people and all the things they're studying. And interestingly enough, like I said, you can't read it online because it's copyrighted. But you can go to Google and like search a word now, and it kind of shows you like maybe a little snippet around that word. So that's why I'm hedging my bets with some of this. And it's like, okay, so some of these, at least as far as I can tell, may be unrelated. Okay. But there's other ones like this Glenn Pump thing, definitely related. I mean, the fact that they even got insipidus in there, tuberculosis in there in the 1964 area is rather interesting at best, at worst. Okay. So, and here's you can access this symposium document online, and here I've just snapshotted the first page for you, but I've read it. And this is them all coming together to say, hey, this is this thing that we got to kill. It's 1965. And we're saying, everybody kill it. Don't look at it. Just kill it. And let's develop some new drugs, some new ways of diagnosing things, and just kill it. And that's really in the introduction. But what I've shown you here is the kind of the table of contents. So you can kind of see what's going on there. And then I talked about kind of who were the people there, kind of where they went on to later, etc. But basically what this did is it established everyone was on the same page. It was opportunistic infection, kill it. Look for what's damaged afterwards. That's pretty damning in my opinion. Like, why wouldn't you want to understand it? And here's what they're saying. Hurley's data on untreated systemic candidosis, which is what they called it back then, showed mortality approaching 100%. And Winter observed, Candida is a better clinician than the doctor. That's a direct quote from the symposium. And he acknowledged that the organism detected disease states physicians had missed. And so what they're saying is that, yeah, like when something gets out of balance, that suddenly candida can show up. And so that's why they're calling it this opportunistic pathogen, because they see it as, well, it detects an opportunity, so it jumps in there. And I think that's kind of the wrong, well, it's definitely the wrong way to look at it, okay? What happens is the system gets out of balance and it shifts, and it's got this primary mode that it's trying to get back into from this southeastern Africa that it used to run in, okay? But right now, because our the way our biology is set up without the suction heart, it cannot complete that programming. So it is now finding new ways to screw with your system, new things like the goiters, for example. I think would be probably something that was along its line that potassium iodide helped kind of clear up. But then what's the downstream impacts of that? That every time you push on it, it's going to push back. La la la. So and then I just say, hey, the cost is measurable. It's people are dying, and we have decided to ignore this, and Homo cannabis will enter into the literature at some point. And I'm going to stop right here and just give you kind of my opinion of things. Okay. Homo cannabis is more efficient than Homo sapiens, just pure out. Okay. You've got increased endurance, increased mental clarity, bacterial resistance, just a lot of things. Now, then you are also more vulnerable in some ways to punctures. Like if I was punctured in the abdomen due to the suction of my heart, it would kill me. You can bleed out a lot easier because your blood volume over time decreases dramatically. So there are things like that. So you wouldn't be able to have like an abdominal surgery ever. It just if you were in this state, but you would be less likely to need it because of the way it's. I think all of the things that are wrong, not all of them, but many of the things that are wrong in today's people, with our morbidly obese, our increase in a lot of other conditions from ADHD to let's say even autism. I don't think you can prove that this isn't related. I think that right now I don't think the science out there can say that. I think the science says we need to look and find out if those are related to the ECS system. And if they are related to the ECS system, then being Homo cannabis would get you back into a thing where those wouldn't be a concern anymore. Now you're going to have these periods of weakness and everything else. The whole medical establishment would have to change. All the institutions of medicine, the big pharma, they're going to fight it. The doctors are going to fight it, etc. But I think AI will take us there. I think AI will take us there. And I think that in the end, it's going to be part of the fourth turning. I don't think it's going to be part of this fourth turning as much. I think this fourth turning, that like all of my science, is too late to really have a big impact. I think, maybe here in some amount of time, some of this comes to the fore and people start really thinking about it. But if you kind of give us, 80 years, where are we then with this knowledge? I think the next fourth turning is prime territory for dealing with what this is going to bring about. And I'm going to talk about that in a paper too. So the next paper I'm actually going to write, I think, is this one on. So this is a pretty interesting article that I found. So they used to have what they called focal infection theory, and so they thought, you can see it's right here, focal infection theory, and this was kind of predominant in the early 20th century. And these people, they were scientists. Okay, now this guy, there's a poor guy in this article whose son went insane, and he was completely convinced that the reason his son went insane was to do with his biome, essentially, like what was going on in his stomach and everything else. And he, attributed it because they had germ theory. They didn't have really fungal theory at all. And so he attributed it to, well, his bacteria must be wrong. So he did stuff like trying to replace the contents of his colon and did surgeries on him, and the kid died, and it's really tragic. But the point is, and he's not the only example of stuff like this. They had other people who they literally pulled all their teeth out, thinking, oh, that's going to keep you from having disease because you won't have this bacteria anymore in your mouth. And I think they were on the right track. Well, no, I mean, they were going the right direction. They were just on the wrong track. How's that sound? So I think if they would have known about C. albicans and had the science that we had today, they would have looked at it. Now that it's interesting when I write this article, and I'm not going to be able to find it right in through here, but so he did some scientific experiments where he like put adrenaline drops into the eyes, and the eyes differentially dilated. So the adrenaline caused differential dilation in the eyes, and different things like that. So these are some of experiments that haven't been redacted around stuff like this. I'll talk about that in the article too. But anyway, so there's other people, like I said, that are involved in things like this. And this kind of, it fell out of favor because people, they couldn't figure out the ways to fix anybody. So you've got somebody who goes insane, or somebody who has sudden mental changes, and they know something is going on. They don't have the literature or even the vocabulary to understand what is really going on behind the scenes. And so that's what science is build on the foundation of other people. And these people are scientists. I applaud their work. It's very tragic, but by knowing that they did this science, okay, you know things that were tried and failed, okay? And you also know that people thought kind of along the same direction. So it's very important to do things like this. So that'll be another article. Yeah. So and then let's see if I've got anything else. I was going to cover a couple of my test results. This is like totally random, but I decided I would go get a couple blood tests this week, and because I'm detecting some changes that I think might show up on a blood test here. But here's the thing: these will be the last blood tests I ever get, and there was just 3 of them, and I don't plan on going back. But here's an interesting one. So I'm going to have a hard time explaining this to you, but absolute eosinophils right here. This is a transition point for me, 2022, and also in 2014 when I was in a transition, I had this same level of absolute eosinophils. Okay, so what we're talking about here is the size. This goes through an electronic machine, and it measures the size of the cells and the electrical charges on the cells, and it sorts them out into these buckets. Okay. And that's all it looks at is the size of cells and the charges on the cells. And you can do a smear where they do the same type of thing, but once again, the dyes and things that they rely on there are looking at the charges of the cells and things like that. So what was said in the original article that I read back in 1995 was that this test, all tests, it would show up as normal or be interpreted as normal, even when they were actually abnormal. And for example, on a CVC, the example they gave was that due to the changes of the charges on the cells themselves and the difference in the gradients between the intracellular and extracellular areas, so this would be like the fluid gradients, that the cells would be a different size than expected, but they would also have a different charge than expected. And so when they would go through this machine, basically it's confusing apples for oranges, and it's just designed that way. And I mean, it's designed that way, people. Like if you knew, and they did know, okay, in 1964, they did know about this science that said, okay, and right about then is when they adopted this machine. I just find that highly suspicious that that they adopt an electronic measuring machine that by definition will never catch this dichotomy, contradiction in things. And even when you look at it manually, it basically is very easy to get confused about what you're looking at because of the charges and the cell sizes being different. The cell sizes has to do with how old they are and things like that. And so having no eosinophils right here, these are, I think, the youngest guys. Okay, so you look here, these are the smallest ones. So these are the youngest guys. Okay, and what this is saying is, yeah, you didn't have any young guys. So I don't know. That's interesting. Here's another just kind of a what goes wrong with medical test type thing. So you can see on this one that my bun creatinine ratio is not displayed. Okay, they didn't. They said the reference range is 6 to 22, and they said not reported because your bun and creatinine are within the reference range. So that's basically saying, hey, you've got A and B, and those are both within the reference range. So we're going to assume A over B is in the reference range, which is mathematically stupidity. And someone actually had to code this. Like some poor developer sat there and was told, ignore the values up here. Don't calculate this value if these other values are in range. Okay, even though if I put 7 in here for urea nitrogen and 1.3 in there for the other, okay, let's look at what that would do. 7 over 1.3, blah, 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 blah. Well, that's one thing, okay? But now then go for 25 over 0.7. Okay, 25 over 0.7 is going to give you something in the 30s, okay? That's not going to be in your reference range, okay? Just pure stupidity, okay? Then here you can see my potassium was alerted as low. Potassium is that could be from diarrhea, easy things to explain. But it can also be your system trying to compensate to make your heart beat less hard. So anyway, and this test was, well, you can see all my personal information. I really don't care. This test was last year, a little over a year ago. And I was going through a lot. So I'm still going through a lot. But you can see everything else, perfectly normal, guys. Did a UA, everything shows up. Oh, we have ketones, right? So oh, you're eating too much protein, or and there's protein in you. So, but these are, these reference ranges are discounted by the current way of looking at medicine. I think they look at one line, they have a one-line explanation for why that thing could be irregular, and they never take into account the whole system, the whole trend over time. And AI is a master of that, especially this bun thing up here. Like if you just show this, if you take these 4 lines and throw them into any AI and say, so what do you think about that? Is that a valid thing to say, that this bun and creatinine ratio within reference range? And they're like, no, that most likely indicates that you're burning proteins and different things like that. And you are. I'm specifically during most of this, you're burning your own substrates, blood proteins and other things. So all right. I think that's pretty much it. I did want to do, here's my general commentary. I got my spicy Thai on today. We got Tabasco, but the situation in the Middle East, the situation in the United States, the situation globally is continuing to decline. And if you've watched all my videos, pretty early on, I start saying, okay, fourth turning is coming. Everything is just going to continue to get worse, and do not expect things to improve. And I think most people have normalcy bias. You have normalcy bias. You think, oh, I've seen high gas prices before. This is going to fix itself, and we're going to go back to the way it was. And oh, AI isn't that big a deal, it's okay if our country is led by a pedophile with a bunch of people around him who are sycophants that realize that he's basically insane. He's just out there, in one 5-minute clip, he will give everybody the blurb that they possibly could want for their channel. He'll give a conservative channel a blurb that says what they want to hear. He'll give the liberal channel a blurb that says what they want to hear, or what they can at least say to their people. And they all contradict each other. But it works for today's, media environment where most of the time they show you 5 seconds of what was said, and you don't see the whole 5 minutes where he literally looks like he's insane. And it's all purposeful. Just yesterday he was saying how, well, we can't afford to take care of daycare. This is a video that when I just go look for the Trump daycare video. And we can't afford to pay for medical care for people. Well, I mean, I would agree with the second one, actually. I think we cannot afford to pay for medical care for people. But I think there are multiple reasons that we cannot afford to pay for medical care, and those are the things that we should be going at. Okay, one of them is redacted science. I think like we have fundamentally designed a system that silos knowledge from other areas and prevents us from seeing something as critical as a symbiote that is in everybody's system, that literally causes evolution. Okay, that is literally the reason we're standing here talking and walking today. And instead, we're producing glyphosate and things like that literally directly attack the ECS system, which is the communication channel, so that thing is trying to communicate with us in our body. So that would be one way. Another way is let's just realize that the whole United States is built on this capitalistic thing where you've got the Cantillon effect, where money is pouring into the people closest to where the money comes from. Okay, so they get so much money that they need to go put it somewhere. All right, and so where do they put it? Well, hopefully Bitcoin eventually, but where they've been putting it is in all these private equity places. Well, those places are blowing up as we speak. Literally over the last couple of weeks, we've started to see these gated withdrawals on these things, because what they've done is they go buy a bunch of veterinary clinics, and then they raise the prices, and they just have to hold on to it for a certain number of years, and then they flip it to somebody else, and that person comes in and they like, oh, now these are basic services that you must do, and so they get a little extra return from it, and they can value it higher and show that that they make more money on it. But what that's doing is it's screwing the middle class just to the floor the whole time. And the same goes for medicine, is that they've come in, they've bought hospitals, they bought equipment makers, they've bought the just everything. And so all they're interested in is return on investment. Okay, and so that's the only thing. And so everybody's got to have their little piece. And when you walk into a hospital and you go get a test, well, there's every single thing you see got paid for, and there's probably 5 people in that chain who had to get their piece of the action, and a lot of that is owned by private equity, or even if it's just equity, they're just looking for their additional cut. And so we have a system designed, okay, to inflate. And as far as health goes, it's obviously important to everybody. So everybody's going to say, well, I want to live, I'll pay anything. Okay, well, how much money did you make in your lifetime? The average person with no college degree makes maybe, I don't know, something $1.5 million, something like that in their lifetime. Let's call it 2 and be generous, I think. Okay. All I hear when I talk to boomers is about their surgeries and their treatments and everything else. You go have a heart attack and one open heart surgery, you just spent $1 million. And that was half of your lifetime income. Okay, so I'm not saying you shouldn't be saved. What I'm saying is that price is not realistic. That there's too much going on in that price. From pay to doctors, to pay to equipment makers, to pay to the hospitals, to pay to the pharmaceuticals, and everything else. It must be redone. Okay. Now, a better way to do that is Homo cannabis. If we can convince society that Homo cannabis is the way to go, I believe many of these expenses go away. Now, how does that change us? Well, we have to develop a new medical system that is designed around homo cannabis, that is designed around keeping people from transitioning when they get to a transition point, monitors, the things that don't get monitored by today's tests. And I think we'll have a more intelligent population, a more in shape population, just everything. I mean, you literally can't gain weight, fat while you're in this condition. It keeps your glucose levels very stable because the candida is there to consume the extra glucose. Okay, so maybe it's worth a shot, folks. I don't know. All I'm doing is, it's my job to try to communicate these things because nobody else is communicating them to you. And I know from my personal experience in my life that all of the science that I'm basing this on was found, was documented, and then was thrown away, was redacted. So that's what I'm trying to do. And I know this was a huge video, and if you made it all the way through it, you are a trooper. I would encourage you to please share my information. I send it to the smartest people you know. Okay, and if you know somebody who's an archaeologist or a paleontologist or a microbiologist or anything, send it to them. And I don't need 100% adoption rate in 5 years. I don't even need 2% adoption rate in 5 years. I think what we need to do is start the conversation, and history will render the verdict. Okay. So, and then you'll have been there for the beginnings of it and said, man, I remember when I watched this video on YouTube or this video, on Odyssey, this guy was talking about this stuff and everybody thought he was nuts. And it turns out that he was pointing at real stuff. So I guess that's kind of my points for today is, lots of papers, lots more papers coming out. I'm enjoying it. I want to say physically, it's hell. Emotionally, it's hell right now. It's existential. I document those moments out on Nostr, and I'm just 100% honest about it. So if you don't like reading, something that is, rather clinical and rather just kind of narrative, whatever, you can skip those. But they're all out on nostr.jimcraddock.com too, so you don't actually have to join Nostr, but you should. It's the future. It's decentralized. And I hope that you share the information. I think me talking to a few people isn't going to get us there. What I need is for the right people to get involved, and I expect at some point serious pushback. But I live an existential life, and I don't really fear the things that those people might be able to bring to bear on me. So I publish this under Redacted Science, not under my university affiliation. That option was given and declined. So we'll see, right? I will say I do enjoy this. I find great meaning in it, even as I endure many things that I would not want to endure, or if I didn't have to. Over these last 4 years, the worst part about it isn't what you have to endure. Okay, the worst part about it is the unpredictability and the existentiality of it, which can be rectified with a scientific program based around it, where okay, we can monitor you, we know what's next. This is, it's a definite progression, and if we embrace it, it takes us to the next level. I think I could be wrong, but I know it. We can embrace it, and I know it can take us someplace different. And I think that science is advancing, and this is one potential avenue for solving a lot of our problems, a lot of our health problems that shouldn't exist, that didn't exist with those groups back then. Now then, maybe we find new ones. I don't know. But I think most of these ones where it's obviously system out of whack type things go away. So once again, this is Jim Craddock. I have terminal onset diabetes insipidus with candidiasis major. It's been redacted. I have unredacted it. You can read my stuff everywhere. Just Google Jim Craddock Redacted Science. And you are welcome to contact me. My contact information is out there. Nobody has done that, so you would be the first. First in line if you want to have a real conversation about it. I'm definitely open to it. I have attempted to contact one of the living people from that list, who's now in his late 80s, and still university faculty. At least he has a university email that still works and doesn't bounce. And I've been tempted to contact his son, who is university faculty or maybe recently retired university faculty. I can't quite tell. And he has also declined to answer my emails. So I think that in and of itself is interesting. But I'm out there doing what I can for everybody else. And I appreciate you listening. And that's it for today.